Okay, so my name is Alice Pinto, and I come from the Polytechnic Institute of Braganza in Portugal. I've been working with Honeybee genetics for now over 20 years, and today I will be presenting very fresh data on honeybee genetics across Europe. Okay, so the research that I'm presenting today was conducted in the framework of European project, Better Bee, and many beekeepers and colleagues from across Europe have been helping with the collections of samples from 1 to 2 colonies per apiary in more than 1,200 apiaries, representing 33 countries and covering the natural range of 9 European subspecies. This map shows the collections in which the red stars, they represent the apiaries that are involved in conservation programs. The number of samples varied from country to country, as shown in this table, with UK being represented by 32 colonies. As mentioned by, I think it was Matt earlier this morning, this is not many colonies, but so we get a very, it's not a fine scale sampling that we are doing. So we are going to get a broad view at the European level. Most of the samples that we collected were examined for mitochondrial DNA and also wing geometric morphometrics. We are developing at the moment the whole genomes for all these samples, but the results that I'm going to present today are only for mitochondrial and for morphometrics because we don't have the data yet. So in fact, we are still doing some analysis of the mitochondrial DNA. So today I will be presenting the results of the mitochondrial as well as the wings. I'm having a problem in passing to the next slide. Sorry, for some reason, I'm gonna. Okay. Okay, so I will present, starting with mitochondrial DNA. We screened the mitochondrial DNA variation using a very popular and well-known intergenic region, the region located between the CO1 and CO2 genes. Everybody knows these regions, people that work with molecular biology of honeybees. This region allows the discrimination of the different lineages that occur in Europe by the presence or the absence of a DNA fragment, as shown in this portion of a sequence alignment. Do you see the arrow? I'm not sure.
Yes.
Okay,
Yes.
great. That's great. Okay, and this is the data that we got from the mitochondrial DNA. As expected, we found the 3 mitochondrial DNA lineages in Europe. The M lineage is represented by the blue. The C lineage is represented by orange, and the African lineage is represented by green. The C lineage is the most ubiquitous in Europe, even in many countries where the native range of the M lineage occurs. So these include countries like Germany, where we only detected our C lineage, Poland, where the great majority of were C lineage, the Baltic countries, the Nordic countries, and the UK as well. The lowest proportion of C lineage introgression was detected in Iberia, also in Ireland, in accordance with what Grace just showed. And also the French samples seem to be well preserved. But this data we have to take to interpret with caution, because most of the colonies come from from conservation apiaries of AMM. So we cannot overinterpret this data. We detected over 50 mitochondrial DNA variants or haplotypes, with highest number belonging to the M lineage, and being the M4 and M71 the most common haplotypes across Europe. The frequency of each haplotype is represented for each country in the map, in these pie charts. Each slice represents a different haplotype. So clearly we see that the Iberian bees and the French bees have the highest number of detected haplotypes, but there were also quite a few haplotypes detected in the UK samples, including one colony of African ancestry. So I was wondering, where does this African haplotype comes from? And I just learned from Mary today that you are importing bees from Rutneri, and sorry, from Malta. They are supposedly of C lineage, but Rutneri is the local bee, and it is an African-derived bee. So probably this is the origin of the importation, but we don't know. We just know that there are colonies of African ancestry now appearing in the UK. In conservation apiaries, most of the colonies were of M lineage ancestry, as expected. They should be if they are protecting AMM. However, there were also a few colonies belonging to the C lineage here in the Netherlands, for example, and most strikingly, 4 of the colonies from the Netherlands were of African lineage ancestry. In the conservation apiaries of Lezo in Denmark or in Switzerland, all the samples are derived from M4, but we see in France, for example, a higher diversity of haplotypes shown here in this list in the conservation apiaries. This bar plot shows the unbiased mitochondrial diversity across countries. This is unbiased because it takes into consideration the differential sample size and corrects for these different sample sizes. The highest values were found in Western European countries. Here we have UK in the 4th place. This is because M lineage is more diverse than C lineage in the intergenic region, but also because of C lineage introgression is bringing in more haplotypes. So naturally, this is a metric, is measuring the number and their abundance of the haplotypes. So this doesn't mean that this is good for the bees. I think, as explained by Jonathan before and Matt, what is important is whether the bees are adapted or not. I will show next the results of the wings. So we collected 5 workers per colony of the same colonies that we look at mitochondrial DNA. And these 5 wings were identified using the software Deep Wings that we developed some time ago. So this slide shows the lineage distribution inferred by Deep Wings, represented by the outer ring, like here. and mitochondrial DNA is the inner ring. As you can see, for the most cases, there was a good agreement between the two markers. However, in the UK, there seems to be a higher proportion of C lineage by the mitochondrial DNA than as inferred from the wing shape. This can be explained by the differential inheritance of both markers. After several backcross with the AMM, the wings they will look like AMM, but the mitochondrial DNA can pass intact of C lineage from generation to generation. And to summarize, so C lineage seems to dominate mitochondrial landscape in Europe, even in many countries of native range of lineage, such as UK, Germany, Poland, Belgium, the Baltic countries, Nordic countries. The lowest C lineage introgression was detected in Iberia and in Ireland. The highest mitochondrial diversity was detected in the countries of the Western Europe. And the conservation apiaries are dominated by M lineage haplotypes, but there were also detected colonies of C and M lineage ancestry. And we got a good agreement between the mitochondrial DNA markers and wing shape, at least at the lineage level. Well, this is it. This is very simple results. But what is new about this study is the scale that has never been done in Europe. Soon we will have more data when we develop the genomes. And well, this is, I'd like to acknowledge the BetterBee for the funding, the European Commission for the funding through the BetterBee project, and many people, colleagues are listed here that contributed with samples. They organized the sampling in their countries, and my students for helping with the analysis. And thank you for your attention. And well, I'll be happy to answer any question in the session. Thank you.